Hello, my name is Jason Lohmueller, and I'm an assistant professor in the Department of Surgery and Immunology at the University of Pittsburgh. And my lab works on engineering chimeric antigen receptor T cells for cancer therapy. And today I'm going to tell you about some of our work on engineering universal CAR therapy. And so for a brief outline, first I'm going to go over background of CAR T cell therapy, and then an introduction to universal CAR T cells, as well as the universal SNAP-CAR T cells that my lab is working on. And then I'm going to go into new directions with universal CAR T cells, universal SynNotch cells, as well as conditional control of universal CAR T cells. And so first off, the general area that we're working on is an area called adoptive T cell therapy for cancer. And the way this therapy works is that a patient goes into the clinic and their PBMCs, or peripheral blood mononuclear cells, are harvested, and then T cells are isolated and then engineered to either make a CAR or a TCR protein. And again, my lab works on CAR engineered T cells. And then the patient undergoes host conditioning chemotherapy, and then the CAR T cells are introduced into the patient to have some kind of anti-tumor activity in the patient. And so what are CAR T cells? So CAR T cells are, again, the patient's T cells that are engineered to make a CAR protein, or a chimeric antigen receptor. This protein has three general components. It has an antigen binding scFv, which recognizes the target protein or antigen on the tumor cell, and then it has different T cell signaling domains within the intracellular part of the CAR. And upon binding to the tumor antigen, the CAR will become activated and lead to tumor cell lysis, and also production of things like cytokines or other gene program responses. And there's a lot of excitement about this therapy, mainly because of the dramatic results that have been seen in the clinic with hematological malignancies. There are now 5 FDA-approved CAR T cell therapies. 4 of those are targeting the CD19 protein on different B-cell cancers, and then the newest is targeting the protein BCMA, also on hematological malignancies. And this original FDA approval was based on really dramatic results with patients who had failed standard therapy, who normally have only a 7% 5-year survival rate. With CAR T cell therapy, these patients now had over 83% complete responses. So in the clinic, the first 2 CAR T cell therapies that were approved were Kymriah and Yescarta. And again, they had really exciting results because, as you can see here, these are survival plots of progression-free survival over a time. And you can see that even at 24 months post-therapy, about half the patients are completely cancer-free. And again, this is much better than traditional therapy. However, there are still 60% of patients or so who are not responding. And so what my lab is working on is trying to address this 60% of patients who are not responding. And so what are the reasons for that lack of response? So there are several limitations that have now been discovered in terms of why the therapy is not working for those patients. And so one of the original ones is that CAR failure can happen at the manufacturing phase. So patients can have T cells that won't expand enough, or they will have limited persistence in the patients. Another mechanism is having antigen modulation. So the patient's cancers can actually lose the targeted antigen, either CD19 or CD20, and or it could get down modulated on the surface, so that CAR T cells are no longer able to target those cancers. And then next, there can also be toxicities from the therapy being so potent that will actually lead to cytokine responses that become toxic to other tissues. And then finally, there's a great unmet need in treating other types of cancer. So currently, these are working for leukemias, but solid tumors really make the majority of the cancer burden around the world. And so to address these the shortcomings, my lab and others are working on developing universal CAR T cells. And so what is a universal CAR? It's a CAR T cell where instead of binding directly to a tumor antigen, the CAR T cells are co-administered with these tumor-targeting adapters or anti-tumor antibodies. And the way the CAR works is that instead of binding directly to the tumor, it binds to the tag on the antibody, and then the antibody binds to the tumor cell. And so why do this? So this therapy potentially has several advantages over current therapy. One of these is that there can be tunable activity. So instead of dosing in different numbers of T cells, what we can do is we can dose in different amounts of the adapter, that anti-tumor tagged antibody. And in dosing more slowly, you could potentially avoid toxicity by ramping up the amount of the anti-tumor activity in the patients. This also allows the clinician to target multiple different antigens or proteins on the same tumor cell by applying different anti-tumor antibodies with the universal CAR T cells, as you can see here in this diagram. And then finally, it's also very exciting because this universal CAR T cell could be used not only for hematological malignancies, but also for solid tumors, simply by combining the universal CAR T cells with different anti-tumor adapters. And so it's a highly versatile therapy compared to traditional therapy. And so again, this is how the therapy would work. So the patient's blood cells are isolated and then transduced with a virus to make the universal CAR. Those cells are then expanded and then reinfused back into the patient. And then at the same time, the patient is administered with that anti-tumor adapter molecule. And this again gives control over, more control over the timing of the therapy as well as the potency as the anti-tumor activity. And to date, there have been several anti-tumor universal CAR T cell therapies developed, and the different variations really come in the type of adapter that's being used with a therapy, as well as the targeting molecule on the CAR. So you can see here they fall into 4 generic categories, and I'm going to tell you about a 5th one that we're developing in our lab. So this first one is taking an antibody that has a natural binding partner on the immune receptor. And so in this case, the tag protein is recognizing a biotin molecule that's attached to the antibody adapter. And then that antibody recognizes the antigen on the tumor cell. Then there are also anti-tag CARs, which recognize other tag molecules placed on the adapter molecule, as well as bispecific engaging immune receptors, and then ADCC mediating immune receptors, which target the constant region on the antibody adapter molecule. And so what we're working on in my lab is developing universal SNAP-CAR T cells. And so these are slightly different from those other adapter CARs that I went over on the last page. And so the way this works is our CAR has as the targeting molecule the protein SNAP. And this is a human enzyme that's engineered to react with benzylguanine. It's a benzylguanine tag that goes on the adapter molecule. And what makes this really unique is that instead of simply binding to the tag, the way that SNAP works is it will actually perform a chemical reaction with the adapter and form a covalent bond with the antibody adapter. And so when the antibody is administered, it will form this covalent interaction, and then it can sense the antigen and lead to T cell activation and effector functions. And so what we do in the lab is we conjugate the benzylguanine tag to the antibody, and we've done this to several different anti-tumor antibodies targeting a variety of antigens, and these are all different clinically approved antibodies that are currently in use. And you can see we're targeting antigens like CD19 and CD20, which are found on blood cancers, as well as EGFR and HER2, which are found on solid tumors. Again, hoping to find that this will be a universal programmable therapy. And so what we did was we made a lentiviral DNA construct encoding the SNAP-CAR, and then we packaged it into lentivirus, and we transduced primary human T cells with the construct. And again, we have that SNAP tag domain, which forms the covalent bond with the adapter. And then we made the rest of the CAR with the hinge domain from the CD8 protein, as well as the 4-1BB and CD3 zeta T cell signaling domains. And we're using a blue fluorescent protein as a marker for cells that were transduced with the CAR. And you can see here on the right flow cytometry plots where we see efficient CAR expression in the T cells. So you can see on the x-axis tag BFP expression, as well as on the y-axis staining with the SNAP surface dye. And this is compared with cells that were not transduced with the virus. And so next we wanted to see, can we use these SNAP-CAR T cells and different anti-tumor antibodies labeled with the Benzylguanine to retarget CAR T cell activity against different cancer cell lines, plus and minus these different tumor antigens. And so we mixed the tumor cells and the T cells together, as well as the different anti-tumor adapters, and we assayed for interferon gamma production. And you can see here that we only saw significant interferon gamma production when we had matched antigen and antibody combinations. So here we're targeting CD20, and then next EGFR, as well as both HER2 and EGFR on an ovarian cancer cell line. And next after this, we want to see, can we actually lyse tumor cells using these universal CAR T cells and adapters? And so in this case, we assayed for two different outputs. We looked at one marker for T cell anti-tumor activity, which is cytotoxic granular release. The marker CD107A becomes upregulated on the T cells. And again, we see a pattern in which only when we have the matched antibody and antigen combination do we have upregulation of CD107A. And then we actually looked at lysis of tumor cells. And here, again, we only see significant lysis of the cells when we have matched antigen and antibody tumor targeting specificity. Okay, and then to really show off and examine the universality of the therapy, we wanted to see, could we use the therapy to actually at the same time target multiple antigens? So this would be what we'd want to do to try to avoid cancer relapse due to antigen loss. So if a tumor has both, for instance, CD20 and EGFR, can we use both antibodies and efficiently target the both antigens at the same time to avoid that loss? Or if we had a tumor with pre-existing variation where some cells only had CD20 and some had only EGFR, could we eliminate all of those cells in addition to the ones that have both? And so what we did was we mixed, we first, again, we're looking at specific lysis. So this is killing of CD20 tumor cells using the rituximab with BG, as well as EGFR expressing cells that have killing of those using a cytotoximab with BG. And we see very efficient and specific killing using those antibodies on the single cell populations. And then when we mix the cells alone, what we see is if on the top plot we have the killing of only the EGFR cells. And so when we add in cetuximab alone, we see efficient killing of those cells, but not of the CD20 positive cells on the bottom plot. And similarly, when we add in rituximab, which targets CD20, we only see significant lysis of the CD20 positive Raji cells, but not of the EGFR cells. And when we combine both antibodies, we see very efficient killing of both target cell populations. So indeed, this could be a good therapy to target multiple antigens at the same time. And so for next steps, we're moving the therapy, we're advancing our development of the therapy to testing in animal models. And I'm not going to go over those experiments today, but I do want to briefly cover the the current status of universal CAR T-cell therapies as they're moving to the clinic. So now, as I mentioned, there are several groups who are developing universal CAR therapies, and they are moving to the clinic. And here you can see several of the different technologies and their current status. And I want to include this also as just a general reference. But there are several trials being undertaken targeting different blood cancers, as well as now some solid tumors using trastuzumab targeting HER2. And so these are very exciting, and we hope to advance the SNAP-CAR T-cell therapy also to trials in the near future. So in general, there are several pros to CAR T-cell therapy, but there are also some limitations, and I just wanted to review those here. So the pros are that universal CARs can target multiple antigens in the same tumor in order to potentially prevent relapse, because you can combine the universal CAR T-cells with multiple adapters to target different antigens at the same time. And so if the cells lost CD19, you could also be targeting CD20 at the same time, for instance. You could also use the same CAR T-cells for both a hematological malignancy as well as a solid tumor, just by combining them with different adapters. And finally, we can also control activity by dosing in different amounts of the adapter. And so for the cons, so it is a more complex therapy. So of course, the therapy now consists of both a cell component and some other adapter component. So that could be more complicated for regulatory reasons. But in general, as we're using already approved antibodies, I think that's less of a concern. There's also some concern with tumor penetration using adapters, because you also need the adapters to potentially penetrate the tumor as well as the T cells. And we're hoping with the SNAP-CAR T cells, as they're covalently labeling the T cells, that this would be less of an issue, because the tumor trafficking of the T cells can be pre-labeled by the antibody and then trafficked to the tumor. Okay, and so now I'm going to switch gears into some next directions that we are undertaking with universal CAR T-cell therapy. And so the first is has to do with a new variety of engineered receptor. This is work out of Wendell Lim's lab in UCSF, as well as Cole Roybal, in developing these SynNotch receptors. And so a traditional CAR, upon binding to an antigen, will activate T-cell effector functions like cytokines, interferon gamma, and target cell killing. But the way that SynNotch receptors work is that they will sense an antigen and then activate any gene or genes of interest in response. So it's a much more programmable kind of designer gene response that they can make. And so the cool thing about this is that you can engineer T cells that would locally deliver a specific payload instead of simply focusing on killing the tumor cells. Maybe a better idea is to have them help out other things, such as different other therapeutic agents, which could either have anti-tumor function or could be used for other types of diseases, as well as controlling the fate of the T cell, and also stimulating endogenous immunity. So can you make some molecules that will activate other immune cells to have anti-disease function? And so what we wanted to do was, could we see if we could make T cells that had universal SynNotch receptors to provide some of the benefits of the universal CARs to the SynNotch receptor technology. And so again, this is the way the SynNotch receptor works. So it takes a novel environmental input and leads to a novel transcriptional output. And so what happens is, upon binding to the target antigen, the receptor will get pulled and then cleaved and release a transcription factor to turn on that designer gene program. And so what we wanted to do was create one using the SNAP protein. So we made a SNAP SynNotch receptor that we would combine with Benzylguanine tagged antibody adapters, and then lead to a covalently assembled receptor. And then downstream, also in the same cell, we have a response gene, which has a promoter sensitive to the transcription factor, and then an output gene. In this case, we're using tag BFP blue fluorescent protein. And so upon antigen binding, the receptor will get cleaved, leading to production of that output gene. And so if indeed, when we add these receptors to T cells and we combine them with or without the adapter, on the left we have an anti-CD19 adapter, and we have them either with CD19 positive or CD19 negative cells, and we assay for tag DFP expression, we see that we are able to redirect SNAP SynNotch activity to produce the output BFP protein only in response to antibody adapter and antigen on the targeted cells. And we can also do this using an anti-CD20 adapter, the rituximab adapter, again seeing only response when we have the CD20 positive cell. And so this is very exciting, and now we're working on applying this technology to lead to production of therapeutic gene outputs to try to enhance CAR T cell therapy using these engineered SynNotch receptors. And so now I'm going to tell you about another new direction that we're looking at, and this is applying conditional control to the universal CAR therapy. And we're looking at both two different types of off-switch control mechanisms. And so one of these is, can we use light to actually control the CAR T cell technology? So can we make a CAR T cell that is controlled by light to actually avoid an anatomical site in the body? And so here you can see we have a brain which has a tumor on one part, but maybe we know that the CAR that we're going to be using, or the antibody, has some off-target activity on another region of the brain. So can we create control where we can shine light on that part of the organ and then protect it from CAR T activity? And then also, could we create a system that maybe would have systemic toxicity, but that we could tune it or switch it off if we see that toxicity in the patient using a small molecule? And so we have a collaboration with the Deiters Lab in chemistry trying to develop this, and they're experts at creating conditional chemistry control. And we have, because we're adding this benzylguanine to the adapter, we're already using chemistry. This is actually a great venue to try out these reactive chemistries to try to get conditional control. And so the way this will work is we'll take our traditional adapter, but instead of being connected, having the benzylguanine connected by an inert linker, it will have a reactive linker that will either get cleaved by light stimulation or by a small molecule addition. And so when the adapter is present and it has no stimulus attached, it will act like any other adapter and activate SNAP-CAR T cell activity. But when light or the small molecule are applied, it will cleave that benzylguanine off and won't lead to any signaling by the therapeutic T cells. Okay, and so working with the Deiters Lab, we created these cleavable off-switch adapter where it's reactive to blue light. So when the blue light is applied to the system, it will cut off the tag molecule on the adapter. And so we combined the off-switch adapters with the universal CAR T cells as well as the target cells. And then we applied light for different amounts of time, and then assayed for tumor cell lysis. And what you can see on the right is that if we had traditional CAR T cells in green and blue, if we applied them to tumor cells, they were unaffected by light stimulus. And if we also used an adapter that had an inert PEG-2 linker, which is our standard linker, in orange, it also was not affected by light. But when we used our light cleavable adapters at two different concentrations, we saw that we get induced suppression of the tumor cell lysis. And so we were able to turn off lysis using the light for these two adapter antibodies. And even at 10 seconds of light exposure, we already saw a dramatic reduction in near complete reduction by 45 seconds of exposure. And so for the clinic, the real excitement comes to applying this where light is already being used, which is in fluorescence-guided surgery. So currently, there are some systems in which patients are applied with a fluorescent contrast agent, and this helps to visualize the tumors in the patients and help guide the surgeons who are performing treatment. And so what we're trying to do is to also create cleavable or activatable linkers that are reactive to other wavelengths of light, so in the near infrared wavelengths, to hopefully use them in this type of therapy. So we also built a drug-controlled off switch. And in this case, we're using a small molecule drug, phosphine, called 2D-PBM. And what we can see on the right is that if we add the drug and the adapter at different concentrations to tumor cells, and then we stain for adapter signaling on the z-axis, you can see we can get very tunable control of the amount of tag on the surface of the tumor cells. And then similarly, when we go to look for control of CAR T-cell tumor cell killing, we also see we get a very nice tunable control over CAR T-cell function. And you can see, again, if we have inert linkers, the PEG2 linkers, or a traditional CAR, we don't see any reduction from the small molecule being added to the co-culture. But if we have our cleavable linkers, we see that nice reduction. And it's close to the no adapter control. And so for the next steps with the conditional control technology, we're also very interested in creating on switches. And I think our general excitement here is that we can provide even more control and more tumor specificity using conditional control. And so we have a few different control stimuli that we're especially interested in. And the first of these is, again, using light. And the really nice thing about using light as an on switch is that instead of protecting some anatomical location, we could actually use light to direct the activity. So if we have an unregulated CAR T-cell therapy in which there's a specific tumor site that we know we want to hit, but we're worried about on-target toxicity in the rest of the organ, we can apply light only to the specific tumor site and get activity only in that region. And similarly, with a small molecule triggered system, we could apply the adapter antibody systemically, wait for it to collect at the tumor site, and then apply the small molecule drug systemically to the patient to get tuned activity and local activity. And lastly, we're also developing switches that are controlled by tumor microenvironment stimuli. So in addition to making different target antigens, the tumor microenvironment is known to have different chemical conditions, including acidosis, so lower pH, expression of different proteases, which also help tumor cells to metastasize, such as MMPs or legumine, and then reactive oxygen species, like hydrogen peroxide. And so the idea is, can we get greater tumor specificity by engineering adapters that are decaged or activated upon these stimuli being added? And again, so we have our adapter that now has the benzylguanine, but this red cage on it. And when we apply these different stimuli, they will lead to decaging of that benzylguanine and then activation of the SNAP receptor signaling. And so this is where we're headed, and I think it's these types of advancements that will lead to universal CAR T-cell therapy being applied to many more types of cancer and ultimately making a dent in the cancer epidemic. And so with that, I want to acknowledge my lab and everyone who contributed to the project and our many collaborators and our current and past funding support. And also thank you for your attention.